So let's see. All right, so the key concepts. Spoiler alert, this is what you're going to take home from this. Cannabinoids, such as THC, mimic the effect of the endocannabinoid system, the system in our body that marijuana interacts with. Mutations of this system, which is nothing more than proteins, is most of this system, and proteins come from DNA. You get a mutation, the protein function is disrupted, which leads to some sorts of diseases. For example, we're all familiar with the appetite-stimulating effects of cannabis, and so if the receptor gains a mutation, you lose the ability to know when to eat or how much to eat. It can result in a disease like anorexia or bulimia. Also, I'd like to talk about some research I've done several years ago about cancer research. I feel like there is a lot of promise and a lot of skepticism here, and we need both imagination and skepticism in order to move forward. So I'd like to talk about some basic research in relating to cannabis and cancer. And I'm going to close with talking about the American Herbal Pharmacopoeia's monograph, which is a regulatory document that was published for the standardization of cannabis. So we're going to talk about some research studies. We're going to acquaint you with the field, a nice overview. Going to go into the basics of the endocannabinoid system, talk a little bit about some benchtop laboratory science, CB1 receptor, the receptor that THC activates. We're going to explore its structure, its function. We're also going to talk about cannabis compounds. I'm going to walk you through my favorite compounds off the cannabis plant, including terpenes. And then I'm going to go into basic product safety for botanical medicine. So the cannabis plant is inherently safe, but commercialization can be dangerous. You know, we talk about the largest recall of cars in the history of the United States happened several months ago. Commercialization can be dangerous. I think anyone in this room could probably grow a couple of tomato plants really well, but if you had to grow 1,000 of them, I think that the quality of those tomato plants would vary greatly. And I'm also going to talk about my work in auditing medical cannabis operations for certification. Because we have things like the American Herbal Pharmacopoeia's monograph. So here we have on the left is the year, followed by the number of studies. So in 1843, there was only one study published. By about the time the Controlled Substances Act was authored, we only had about 200 studies. In green, you see in the 90s was when the scientific community discovered the endocannabinoid system. The receptors were officially identified, their genes were known, what genes encode these receptors, as well as the discovery of endogenous compounds, compounds within our body that activate these receptors. So we have this receptor system in our body, and there's this plant out there. But we don't have the receptors because there's a plant out there. That is a quirk of nature. We have the receptors for THC in our body because it's part of an important system. Just like opiates and endorphins, endorphins are endogenous opiates that your body makes. So we have many of these compounds in our body. But what I'd like to point out is that in 2014, about 2 months ago, in the searches, I found over 1,500 research articles published this year alone, internationally, on cannabis and cannabinoids. So when someone says, oh, we need more research on that, I find that to be an incredibly unuseful thing to say, because I have not even had time to scratch like 5% of those studies. So when someone says that to you, ask them how many studies have they read this year. Usually, you know, there's no department of any government, local government, that's tasked with reading these. They just say there's need for more research, which we do have some questions like, how well does it treat diabetes? Not if it can treat the symptoms of diabetes. You know, how well can it treat spastic disorders and things like that. So the endocannabinoid system. The cannabis plant has been a medicine for millennia with unparalleled versatility, but its mechanism of action remained a grand mystery since the plant was known. And it wasn't until, like I said, THC was isolated in the 60s, which led to people being able to synthesize it and use it in research studies, and in the 90s with the discovery of the receptors. We just are finding out about how this works. But the cannabis plant, it turns out, isn't the only plant to make cannabinoids. It's the only plant that makes THC and CBD, cannabidiol, but many other plants produce cannabinoids, such as Bocanga africana, which is the tree ibogaine comes from, if you're hip to that. Rhododendrons also produce some cannabinoids, species of liverwort moss, as well as some species of chrysanthemums in South Africa have been reported to all produce cannabinoids. And there's actually one cannabinoid slash terpene, a smelly molecule called beta-carotene, which I'll talk about, which is actually found in nearly every plant. If you ate a salad, you ate this cannabinoid. It's ubiquitous throughout the plant system. And so this endocannabinoid system that we have in our body consists of anandamide and 2-AG, which are these lipid-like signaling molecules that THC mimics. Cannabinoid receptors, we have a lot of receptors. The two main ones are called CB1 and CB2. The CB1 receptor protein is probably the most abundant protein in the human brain. There hasn't really been anything shown to be more abundant than cannabinoid receptors in the brain. We are wired for a positive reaction with the plant. The CB2 receptors are found mostly throughout the immune system and help to regulate inflammation. And you also have enzymes, other particular specialized machines, that synthesize and break down the endogenous cannabinoids. And so what does this system do in our body, this elaborate, intricate system? It helps us to eat, sleep, relax, forget, and protect, which is a phrase coined by Vincenzo de Marzo and Raphael Mechoulam. And so eat, stimulation of appetite. It helps to regulate satiety and anti-inflammatory effects, sleeping, relaxing. It also helps us forget, which is just as important as remembering. If I remembered every face I saw from Brooklyn to Williamsburg, my head would explode. You need a system in your body that can tell you what information to get rid of, what is important and what is harmful. And we can see this in many, many conditions that people are trying to treat with cannabis. OCD, PTSD, these conditions in which there's information that is no longer useful or harmful for the individual and they can't forget it. And also protect, which I think is the most important aspect. We know now that cannabinoids, especially plant cannabinoids, do not harm healthy tissue. They actually protect them. THC, cannabidiol, apart from their receptor mechanisms, are wonderful antioxidants with tremendous antioxidant potential. And so they do not harm healthy tissue. They actually keep it alive. And this is also why people are so attracted to these as anti-cancer agents, is because they leave healthy tissue alone and only seem to go after bad tissue. The clinical endocannabinoid deficiency was a term coined in 2004 by Ethan Russo, who's perhaps the real lead cannabis researcher. He's an amazing guy. If you've ever want to have some great reads throughout the decades about cannabis literature, he started off studying the effect of marijuana on Moroccan fishermen's eyesight at night. But the clinical endocannabinoid deficiency syndrome basically describes why certain treatment-resistant conditions can be treated with cannabis. Because if, let me give you an example. If someone has a mutation or a disorder where their appetite is messed up, right? They're not getting the normal level signals to tell them how much to eat and when to eat. Whereas if you stimulate that system in a healthy person, right? You stimulate that system, they eat everything in the fridge. But someone who doesn't have an appetite, you stimulate them with cannabinoids or supplement them with cannabinoids, they're just brought up to the level that they're supposed to be at. So that's why it works so well for people going through a wasting disorder. They're not getting those signals telling them to eat. When you stimulate their system, they're just eating what a normal person would eat. So I think it's important when we think about what we're doing and how we're treating this. I think the endocannabinoid system is extremely important. I believe that our future to treat new conditions, to treat diseases and potentially cure them, depends heavily on our ability to understand this system as well as being able to develop treatments. And the clinical endocannabinoid deficiency syndrome sounds a little silly. You're pot deficient, man. Like it sounds a little crazy, but we actually have a fair amount of evidence. On the left are codes for the CB1 receptor and the CB2 receptor. These are the descriptions of the mutations in horrible technical jargon. But the disease associations you can see. You have a messed up CB1 receptor in animals. They see schizophrenia, they see Parkinson's disease increasing, anxiety, depression, eating disorders, obesity, all these sorts of things related to cannabinoid receptor mutations. And that some researchers have taken these receptors, these mutations, and test them in petri dish assays and found that actually the pharmacology has changed. What drugs the receptors prefer is changed. Some of the work that I was involved with and for my thesis was studying the role of the endocannabinoid system in bone. And what got me interested in that was that in 3 distinct human populations, Japan, France, and Eastern Europe, they found mutations in CB2 receptors, the immune cannabinoid receptor. THC also activates this receptor. But they found that if you had this mutation, you had lower bone mineral density and more severe age-related osteoporosis, which implies that if the receptor is losing function through a mutation, then stimulating it might actually improve bone formation. So I just want to drone a little bit more about cannabinoid receptor structure and what it is, because I really think it's helpful to demystify it, and I think it's one of the coolest proteins out there. And so this is perhaps the most basic model of a cannabinoid receptor or a G protein coupled receptor. This is the cell membrane, or the cell, the outside of a cell. And then it spans the membrane 7 times, going in and out and in and out. And this is where the drug docks. And this is, it conveys its signal here. But proteins are not these simple beads on a string. They're complex bundles joined together by electrostatic interactions, because all of this is made up of proteins and amino acids. This is a protein made of amino acids. And what it looks like to researchers when they put the data into computer simulation is basically this. This is perhaps the grandest scale we know of the cannabinoid receptor. This is probably the last thing THC sees before it activates it. But it's a nice little machine. The drug docks, this little electrostatic bridge breaks apart, and the receptor moves to convey a signal. But then again, you have to remember that these represent amino acids, all these different letters. So this is basically the structure of the protein. Is its nice little bundle, this pocket for things like THC to activate in. And you might see that there's some red letters, like the D and the K. Those are mutations that I made in my thesis to try and understand how drugs are binding to this receptor, and then finding out which ones are important for what aspect. But the take-home message here is even small changes in a single amino acid can greatly change what the receptor is doing and its ability to be stimulated by THC. And so you take this information, you know this receptor's shape, you know its structure, you know its function, you know what drugs activate it. Well, what can you do with that? Well, one of the things that we got to do with that was throw these compounds onto cancer cells. And so we worked with a neurosurgeon who did many surgeries a week, and we were able to treat these cells with THC and CBD. But what I'm going to show you here is a YouTube video we posted. So on the left side are normal, healthy brain cells, astroglial cells, and on the right are the tumor equivalents, and they're both given the same amount of THC. Let's see, and so the healthy cells on the left stay alive, and the ones on the right start to shrivel up into little white dots. So as an undergraduate, I found this incredibly exciting to come into the lab every day and watch videos of brain cancer cells, aggressive brain cancer cells, being inhibited by cannabinoids. And so we took that work, and what we did with it was we started doing combinations. So people talk a lot about plant wisdom, plant synergy. What we did is we took CBD and THC, and we combined them. And so this first column is no drug, and then just THC, just CBD, and then in combinations together. And what we found in terms of aggressive grade 4 brain cancers in Petri dish assays, and I stress Petri dish assays because there are some subtle differences between a Petri dish and a human being you may have noticed. But it's fairly impressive. And so here's a little snapshot of the Petri dish, you have, after 3-4 days, it's completely full. With THC and CBD in a 1-to-4 ratio, you see almost a complete inhibition of growth. These findings were published in the Journal of Molecular Cancer Therapeutics. And so far as I know, is the only documentation of cannabis synergy between compounds, because we actually measured the greater than additive effects that CBD has on THC's effects. Long-term use, but after this I'm going to go into sort of a rapid-fire, flashcard style, fun tour of cannabinoids. But nothing really upsets me more than misinformation. And so the two studies I put up there, Pope and Grant. Pope, 2001, I think is the most important long-term study. This study is the longest study ever conducted on cannabis use in terms of abuse. In order to be included in this study that measured neuropsychomotor performance, memory, things like this in cannabis users, looking for deficits in long-term cannabis users, you had to have used cannabis a minimum of 5,000 times and be a current user. And they measured you with a battery of tests, like I said, looking at memory and cognitive processing. And what they found, regardless of the amount or intensity of use, 30 days after abstinence, there were no difference from controls. Brain scans, all that stuff. And that's the fatal flaw when you look at like studies coming out of New Zealand, is there snapshots of a dynamic process. They follow people for a week. There's no washout period. And you might hear this study about New Zealand quoted over and over and over again. But remember, it's the one about the 8 IQ points, the loss of 8 IQ points. You've heard of this one? Yes. Okay, so first of all, there was no dose dependency, right? They didn't track how much people were using. So there's no correlation between small amounts, large amounts of cannabis. They did not control for binge drinking. They just asked people if they drank alcohol. They took into account schizophrenia, but not any other mental illness or traumatic injury. So if you've had 6 concussions, didn't catch it in their survey. And all their effects were negated by socioeconomic factors of their participants. And there were no correlations. And this is another weird thing, is that they actually found that cannabis users had a denser hippocampus. And usually when you have a denser part of the brain, in all other fields of study, a denser part of the brain is a good thing. More neuronal connections is correlated to things like knowing multiple languages. Like taxi drivers in New York have very, very, very dense hippocampi, because they have to know so much. But yet in this study, that whole good brain, bad brain narrative gets turned on its head. Symptoms of epilepsy, and it can have, in some people who are hyperactive, it can have sedative effects. CBD can also reduce the appetite stimulating effects of cannabis in different amounts. And then in Sativex, which I like to use because it's really a whole plant extract. I'm not sure if you guys are familiar with Sativex. It's from a pharmaceutical company called GW Pharma. It's basically cannabis without any plant matter. It's just this plant cellulose gets in the way, so they remove that, and it has terpenes and cannabinoids. And so they've been done a lot of human studies, and they've shown that Sativex compared to THC, if you have all the components, essentially the whole plant, you can greatly enhance the effects of THC for pain relief, for cancer-related pain, and also with psychomotor performance. One of the things people talk about too is coordination when people are intoxicated on cannabis. Now, perhaps I talked in the very beginning about healthy versus a sick person and stimulation of the system, right? The other one was the healthy person uses cannabis, they eat everything in the fridge, where a sick person uses cannabis, they get to the right level of stimulation. We can also think about that for driving. So if someone has uncontrollable muscle spasms or something like that, and you give them cannabis and they stop, they're going to be a much better driver than they were without cannabis, right? Because if you're driving along the road and you're not constantly making these motions, you might be a safer driver. Whereas early studies looking at driving cannabis in healthy users have mostly showed that you drive slower to compensate for the impairment. But what my favorite part of those driving studies is the stoplight test. And so they drive this little course, and there's a stoplight. And across user groups, the cannabis users respond the quickest to a light changing from red to green. And this, they think, is because there's a subjective change in the perception of how time passes. So while someone who has just consumed some cannabis or THC is sitting at a light, they're constantly thinking, shouldn't it have changed already? Why hasn't it changed? It's been forever. So they're just more primed for it. And they see science, there's another scientist saying this. So CBG, cannabidiol, is another compound of interest. A lot of these slides were prepared with the help of Ethan Russo, who's a scientist. He actually originally lent me these slides to talk to high school students about cannabis. But CBG is a potent GABA uptake inhibitor, which is important for regulating sleep and being more like an antidepressant medication. It's actually sort of the stem cell of cannabinoids. It's produced very early on in the trichomes, and it becomes, depending on the gene expression of the plant, THC or CBD. And so you might see a lot of this in an early plant. CBN is the degradation product of THC, so you leave cannabis in the sun, the THC will convert to CBN, which is basically a very useful compound, but less potent than THC, less efficient. Lots of fun things. It actually seems to be good for the skin, inhibits a keratinocyte proliferation, as well as is a potent antibiotic. These are some terpenes. I'm going to go kind of quickly, but just to kind of tell you that the terpenes, 120 so terpenes found on the cannabis plant are found in other plants as well. And terpenes are, well, they're very small compounds, and you might think, yeah, lemons smell great, but what's the point? Well, the point is that those are active ingredients that you are inhaling, and they are potent at ambient air temperatures. They've actually wafted this compound into psychiatric wards in studies and found that the scent of lemons actually reduces medication use because it has its own antidepressant-like effects. And these are absolutely potent things at very, very small amounts. Myrcene is another one you might be familiar with. It's very sedating, and some of these compounds can interact with THC. You might there's an opiate shortage worldwide. The United States are really lucky, in a sense, to have so many opiates because, like in Mexico, a friend of mine went there, visited a cancer ward, and they treat patients generally with aspirin for their cancer pain because there aren't enough opiates to go around. So one of the things cannabis could also do is provide a way to make sure there's enough opiates for irretractable pain, because if you combine them, you can get a much better effect. Also in lower opiate overdose statistics in Colorado, we're already seeing this sort of play out. And also in rats, one of my favorite studies is adolescent rats exposed to THC, like a ton of THC, later in life are actually resistant to developing opiate dependence when given free access to opiates. All right, so I want to transition to product safety, because with the cannabis plant, we have some issues in the industry. So the first one on the left, we see these two pigeons, one's ripped, and it says, I don't know, I didn't know they were steroids, my trainer told me they were peanuts. So we have to worry about contaminants in cannabis. Are people spiking the edibles with things, or do they have what they say they have? It's an important consideration to think about, like what is causing this effect? Is it really the cannabis, or is it mixed with something else? Also, the consistency, the standardization of medicine, where, as in this cartoon, he says, as soon as the anesthesiologist gets here, we'll get started. I guess back in caveman days, one of the things you had to worry about were the variation, the size of the club, and the guy who was wielding it. So similar, we have to worry about standardization of care in cannabis facilities. So, how are people being consulted? What sort of access do they have? And just sort of a standardization of medical cannabis. And the other thing is accurate product labeling, like having, I can't stress this enough, this is really, really important. We're seeing fallout from this now. I saw a wrapper from a candy bar, I won't say from which state, but it had the amount of milligrams of THC and CBD, and also the percents of THC, CBD, said it was organic, pesticide-free, and on the back of it, it said this product has not been tested by any laboratory. So product labeling, I think, is incredibly important, especially for sensitive issues like GMOs. And we may not be able to identify GMOs in the supply just by looking at them. And so I'm going to go into a brief history of cannabis and talk about standardization of this plant material, where we are today. I'm going to talk about my work with patient-focused certification, auditing medical cannabis operations for certification. So, brief history. 1996, first laws passed in California allowing some protection for medical cannabis users. It was basically meant if you were dying of some disease, you got caught with cannabis, you probably wouldn't go to jail. In 2004, ASA and patients passed the first distribution laws in the Bay Area, so first legal sales of medical cannabis. And then in 2010, Colorado creates commercial distribution. And we need product safety guidelines and product safety standardization because, like I said, while the cannabis plant is inherently safe, commercialization can be dangerous. And so, the American Herbal Pharmacopoeia was published and authored by several people, including myself, but also Lyle Craker, Mahmoud Al-Soli from the University of Mississippi. And this is a standard for laboratory methods, for microscopic identification of different parts of the plant, acceptable limits of pesticides and solvents, anything you would need to know to produce a standardized botanical medicine for cannabis. And I'd like to draw your attention to the cover, because I think the cover of it, this picture of the farm at University of Mississippi, which is where the government grows its cannabis. This picture just sums up perfectly where we are with cannabis today. First of all, it looks like healthy cannabis, but it's growing in the worst possible medium for cannabis, which is clay. It is surrounded by a 15-foot high barbed wire fence, complete with a guard tower and cameras. And this is, of course, to ensure that none of the cannabis plants escape. And so this regulatory document is actually cited in the law in 6 states, including Washington and Nevada, as the guideline. Gone are the days of New Jersey's THC caps. The governments cite this for their botanical safety guidelines. And then there's another player in this, the American Herbal Products Association, APA, which represents about 400 herbal products and botanical medicine manufacturers and distributors, came out with guidelines that it worked on with people from the cannabis industry. So there are best business practices for cultivation and processing, manufacturing, dispensing, and laboratory practices. These are all free and available at appa.org. The cannabis committees are ongoing. These are fluid documents. If you'd like to join the committee, learn about what's going on, you can make changes to these documents. So, like I said, then you have the American Herbal Pharmacopoeia, which is basically purity and laboratory testing methods. Herbal-ahp.net, you can get a copy of it there. And then you have patient-focused certification. These are little seals that go on different products that come from either laboratories or manufacturers. And what's great about this is that doctors in most states cannot tell a patient where to get it, where to find cannabis, what safe place to go, but they can tell them to look for a seal or a QR code or something like that. But this is one of those safety and oversight programs to help ensure compliance with local laws. It involves a physical audit, so I review all their paperwork when I go there and make sure they're following all the SOPs, the standard operating procedures. Document staff training, as well as an annual surprise audit. You can find more out about this program at Patient-Focused Certification. I've done over 6 states this year. I've been to over a dozen facilities. And so patient-focused certification is a nonprofit third-party certification process. And this is how regulations work. A lot of people talk about the U.S. Pharmacopoeia, the USP, but I'm not sure how many people here know that actually the USP, which a lot of people cite, is not a governmental organization. It's a third-party group that makes recommendations to regulators. And another thing about the USP, a little sidetrack and then I'm almost done, but we hear this statistic a lot about how in the 40s cannabis was removed from the U.S. Pharmacopoeia, but that wasn't some brazen move to get rid of cannabis. There was over 100 other plants that were removed at the time from the U.S. Pharmacopoeia. All sort of things, let's see, organic certification, kosher certification are all punted by the FDA to third parties, non-governmental groups. Something to think about. But this is run through the Americans for Safe Access Foundation. And it's based on these APA and AHP criteria. It will also offer online courses for training from the Cannabis Training Institute. So I'm just going to talk really quick about some results from the auditing. And some of the things I've noticed is that state laws can actually interfere with basic product safety. So for instance, in Washington, D.C., it is only legal for a patient to take the cannabis from a dispensary home. They cannot take it anywhere else, even whether or not it's open or closed. So if you get your product home and it's defective, there's moldy, it's not the right thing, it is illegal for you to return it, basically. And another thing, waste disposal. So a little joke here is I used to wonder if 420 was police code for marijuana. But I think police code for marijuana is simply I'll take that. Because when it comes to waste disposal, there's Washington, D.C., again, cops will come and pick up the cannabis waste from the dispensaries, but the dispensaries do not get a receipt of how much cannabis was taken and how much reached the station and what happened to it once it got to the police station. Did it go to an evidence locker? Was it quickly burned? We don't know. And the rules and requirements also differ from state to state. You know, most states recognize HIV/AIDS as a qualifying condition. Pennsylvania almost passed a law this year, but HIV/AIDS and glaucoma would not be a qualifying condition. We all know about New Jersey. No chronic pain is allowed there to treat cannabis, or from the other way around. But so the laws and requirements differ from state to state, which makes it a little bit difficult. But the three biggest corrective actions I see when I'm out there are adverse event recording and reporting, recall plans, and security issues. So just keeping track of people, if they report any negative effects, and whether or not a recall will be warranted. Having a system in place to determine that is really important. So what does this QC process look like? I have like 2 slides left. Just to lift the lid off it, this is GW Pharmaceuticals' sort of slide about how they prepare cannabis. So they have their THC and CBD varieties here. They're cured. They're tested for potency and contaminants. They go into a giant supercritical CO2 extraction machine. They're tested before and after. They get this nice sticky tar-like residue, which is then tested before some mint is added for flavoring and palatability. And it's tested again once it's bottled to ensure that the quality is maintained. So it just involves more checkpoints than you might think. This is very important. So just to kind of take the lid off of what a quality control process may look like. And so I guess the purpose of this slide is just to say that cannabis can be grown safely like any other agricultural product. That's the whole point of the American Herbal Pharmacopoeia's monograph and the APA guidelines. You can see that cannabis is growing very fine next to several other plant species. So, in summary, the endocannabinoid system: eat, sleep, relax, forget, and protect. Cannabinoids, while people say that they might be neurotoxic, you have to remember that cannabinoids are prescribed to treat neurodegenerative disorders, not cause them. Cannabis is inherently safe, commercialization is dangerous, and third-party certification of any type will help to ensure basic product safety. And with that, I'd like to thank you, and I'd be happy to answer any questions. Yeah.
So give him a huge that route, and I'm doing that because I'm assuming you'd like to eat while